Hello, and thank you for joining today's Clinical Omics webinar, Detection of Ovarian Cancer Recurrence, Quality Control Routines for Longitudinal Studies using an Integrated Proteomics Approach. I am Chris Anderson, the Editor-in-Chief of Clinical Omics, and I will be your moderator for today's webinar. Ovarian cancer is the leading cause of death from gynecological cancer in North America, and the fifth most common cancer among women. In the United States alone, more than 22,000 women will receive a new diagnosis of ovarian cancer, and it will result in approximately 14,000 deaths this year. Today's presentation will detail recent research using a shotgun proteomics approach using mass spec to identify peptide biomarkers indicative of ovarian cancer recurrence. Our speaker today is Thomas Kisslinger, Senior Scientist at the Princess Margaret Cancer Centre in Toronto, Canada, and a professor at the University of Toronto's Department of Medical Biophysics. Dr. Kisslinger's work focuses on developing an integrated systems biology approach to better understand the molecular mechanisms associated with early ovarian cancer using a mass spectrometer to generate snapshots of proteins present in ovarian cancer cells and tissues. At the end of today's presentation, we will conduct a question and answer session with our speaker. Questions can be submitted at any time. To do so, simply type your question in the panel at the left and hit submit. And now to the presentation. The floor is yours, Dr. Kisslinger.
Okay, my name is Thomas Kisslinger. Thank you very much for inviting me for this webinar, and I will go right into it so we have some time at the end for Q&As. So what I will talk about today, so we'll give a quick introduction to ovarian cancer, background treatment, and current clinical biomarkers that are in use. I will then show you a project that is ongoing, unpublished, and unfinished that was aimed at identifying new biomarkers for early detection of recurrence of high-grade serous ovarian cancer. This project was divided in multiple phases. In the beginning, we had a discovery phase where we used patient-derived xenograft models and a chemical proteomics approach aimed to specifically capture N-glycopeptides. This approach was then followed by a data mining phase, which was aimed at selecting a large number of candidate biomarkers, which were then moved into phase 3 optimization, where we used synthetic, stable isotope-labeled peptides as standards to develop a large number of PRM-MS targeted proteomics assays. These assays were then applied to an evaluation phase, which contained longitudinally collected human serum samples from high-grade serous ovarian cancer patients. In cohort 1, we used a large number of these assays in what we call a PRM-MS screen to relatively quantify these peptides longitudinally. We then mined these data by comparing to ELISA data from an FDA-approved biomarker CA125. We mined these data by slopes, and we selected a smaller number of peptides, which we then synthesized as highly purified aqua peptides for absolute quantification in a second cohort of longitudinally collected human serum samples. So here a bit of a background on ovarian cancer. So epithelial ovarian cancers are the most common ovarian malignancies in women. And they consist of 5 different histological subtypes. You can see this here in that pie chart on the right-hand side. And these histological subtypes can all further be subdivided in many different molecular subtypes. And this is based on sequencing data from large consortia, for example, like the TCGA. High-grade serous ovarian cancer is the most common, most aggressive subtype of ovarian cancers and the interest of my lab. High-grade serous ovarian cancers start, so the cell of origin is the fallopian tube epithelial cells. And early in the progression of this disease, small clusters of transformed fallopian tube epithelial cells leave their primary site and colonize distant sites in the abdominal cavity. So it's at this stage, very early, could be considered a metastasis. These transformed cells colonize particularly tissues that are high in fat, for example, the omentum. And the problem is that at this stage, this cancer is very silent. So as a result of this, most women come to the clinic in an advanced stage where there's massive tumor load all over the abdominal cavity, and many of these women also present with leaders of ascites fluid in their abdominal cavity. Here in this picture, you can see what a high-grade serous ovarian cancer patient looks like at presentation, with many different cancer foci at many different sites throughout the abdominal cavity. Treatment or management of this disease has not changed dramatically over the last decades. In general, it starts with a very aggressive debulking surgery, which is followed by adjuvant chemotherapy. And even these chemotherapeutics have not changed much in the last decades, the platinum-based chemotherapies, even though new therapies are now moving into the clinic, such as immunotherapies or PARP inhibitors. Post-surgery, an FDA-approved biomarker, an ELISA for an FDA-approved biomarker against CA125 or MUC16 is used to measure decline of this biomarker. So one can see this here in this cartoon on the right-hand side. Following surgery and chemotherapy, CA125 ELISA data drops dramatically, or is called normalizes following surgery and chemotherapy. The problem is that most of these patients recur within 2 years. They usually recur with a chemo-resistant disease, and as a result of this, the 5-year survival has not changed very much in the last decades and is very low at 10 to 30%. So a goal of our study, so 5 is a very nice marker. It's not perfect. 15% of patients are CA125 negative, and the sensitivity of CA125 is not perfect. So the goal of our study is, can we find additional markers that would quantify this biochemical recurrence, or this rise following chemotherapy earlier, which could potentially allow additional lines of treatments and hence improve survival of these women with high-grade serous ovarian cancers. Here's the strategy we used for the discovery of new biomarkers. So we made use of patient-derived xenograft models that were originally developed by Ben Neal and Laurie Ailes' lab, as shown in this paper here in PNAS. In that paper, surgically resected tumor specimens were engrafted into the mammary fat pad of immunocompromised animals. So we now took viably frozen surgical resections and reestablished some of these xenograft models. So instead of engrafting into the mammary fat pad, we IP injected viable single-cell suspension of surgically resected high-grade serous ovarian cancer specimens IP into NSG immunocompromised animals. So this even better recapitulates this disease, but has a much lower take rate and a much smaller growth kinetics compared to the mammary fat pad engraftment. Nevertheless, we were able to establish 12 different xenograft models. And at presentation, they look very similar to a human ovarian cancer patient, with tumor nodules all over the abdominal cavity, and most of these animals also present with ascites. We, at presentation, surgically resected PDX tumor as well as collected serum from these engrafted animals. As control, we collected serum samples from unengrafted NSG animals. We then performed a TFE-assisted protein lysis, and at this very early stage, we spiked a yeast glycoprotein called invertase at equal amounts into every sample. So that's kind of like a processing control that allows us to track us how reproducible our sample preparation actually is. We then go through a standard shotgun proteomics sample preparation that digests proteins with trypsin to peptides. We are then particularly interested in N-glycopeptides. The reason is that most FDA-approved protein biomarkers are N-glycosylated proteins, and N-glycoproteins are heavily enriched and secreted and membrane-decorated proteins. To identify these to enrich and identify these N-glycosylated peptides, we used a chemical proteomics approach, which oxidizes glycans with periodats, or cis-diols in glycans with periodat to aldehydes. These aldehydes are then covalently coupled to magnetic hydrazide beads, forming hydrazones. This allows us to very rigorously wash these beads and remove all non-glycosylated tryptic peptides. And estimated this makes up about 80% of the tryptic peptidome. We can then release glycosylated peptides using an enzyme, PNGSF, that cuts between the asparagine and the first sugar residue. This converts asparagine to aspartic acid, which introduces a 1 Dalton mass shift. Within a consensus sequence, we can use the mass spectrometer to identify this. We then perform classic shotgun proteomics using nanoflow UPLC on 50 centimeter nanoflow liquid chromatography columns coupled to a Q-exactive mass spectrometer. We then compare these 3 different sample types, and we're particularly interested in a given quadrant in this Venn diagram, which I will show you in the next slide. We then select a large number of putative candidates, synthesize them as stable isotope labeled standards, and very systematically build targeted assays based on parallel reaction monitoring mass spectrometry. We then apply these assays to 2 longitudinal cohorts of high-grade serous ovarian cancer serum samples. Here's a summary of the results. So in total, we identified close to 4,000 N-glycopeptides that map to just over 2,000 gene products. And in that Venn diagram, you can see how these different samples compare to each other. So here's a bit of a global overview of the glycoproteome that we identified as part of this study. So in total, there were 5,300 deamination sites detected in our shotgun proteomics data, of which about 75%, or 4,050, contained a serine or threonine in the +2 site to the asparagine. So the consensus sequence for N-glycosylation is asparagine, followed by any amino acid but proline, followed by a serine or threonine. So that suggests that about 75% of our peptides were formerly N-glycosylated. These map to 3,779 unique N-glycosylated peptides that then further map to just over 2,200 glycoproteins, suggesting that our approach, is nicely specific. These 2,247 proteins were then mapped to UniProt annotation terms. About 1,800 could be mapped. And here one can see to what subcellular localization these proteins map within our data. So the vast majority, 1,263 or 604, so that's a non-redundant set, mapped to either plasma membrane or to extracellular secreted proteins. Small number intracellular. Many of these are, by annotation, at least members of the Golgi. And here, somewhat poorly visible, is a gene ontology enrichment analysis. On the left-hand side, one can see enriched UniProt keywords. On the right-hand side, various gene ontology terms: biological process, cellular component, or molecular function. The dot plots, the background, the more dark that background, the more significantly enriched these terms are compared to a UniProt background database. The bigger the circle, the more enriched. Red means it's an enriched term. Blue means it's a depleted term. And if one takes a careful look, our glycoproteome is heavily enriched in cell surface, extracellular membrane, extracellular matrix type ontology annotation terms, but is depleted in nuclear or intracellular terms, as one would expect for an enrichment of N-glycoproteins. Then one nice little feature of patient-derived xenograft tumors is that they are mixed species tissues. So here one can actually see an H&E staining of one of our xenograft tumors. So the engrafted cancer cells are of human origin, so they're surgically resected from an actual patient, versus the rest of the mouse, the stroma, is of mouse origin. So one can now use bioinformatic analysis to map identified glycopeptide sequences to either a human or a mouse origin. And here's an overview of how we did it, and I will go in a bit more details in the next couple of slides. So for these 493 glycopeptides, so these are from our quadrant we are most interested in this Venn diagram. So these are peptides that are found in the serum as well as in the tumor of these PDX animals, but were never found in the serum of unengrafted animals. These 493 are represented on the right-hand side as this type of scatter plot. On the x-axis, one has the fold chain, the log2 fold chain ratio of peptide intensity in the serum of the PDX model divided by the tumor of the PDX models. In black, one can see the invertase peptides that he spiked in at the very beginning. They're close to zero, suggesting that our approach is relatively reproducible. If we now map species to these 493 peptides, one can see that 257 uniquely only ever map to a human peptide sequence in an annotated database. 83 are conserved between the two species, and 153 map to mouse protein sequences. Within the set of human unique peptides, we find the current FDA-approved biomarkers C125 or MUC16, mesothelin, as well as an emerging imaging marker called folate receptor. Okay, so how did we actually do this in quick terms? How did we map these peptides back to species? So we used a classic shotgun proteomics approach where we concatenated a human FASTA protein sequence database with a mouse FASTA formatted protein sequence databases. We inverted all these protein sequences into a reverse database and concatenated them also to this database. We then searched our data against this combined database in a classic target decoy approach, which allows us to estimate our false discovery rate, which was below 1%, but then once we identify peptides, we can map them to either human or mouse sequence in this database. And you can see the results of this. So of the 4,047 quantified glycopeptides, 1,769 mapped uniquely to a human peptide sequence in the database that we used for our original searches, which is highlighted in bold here. So we use databases of canonical and isoforms containing about 42,000 human protein sequences and about 24,000 mouse protein sequences. Now, if we take our peptides and compare them into different databases of increasing complexity, and protein sequence databases, they're most definitely not perfect, and assigning species, of course, is associated with a caveat based on the quality of these protein sequence databases. So we compare, for example, to a slightly smaller protein sequence database containing only canonical sequences. 5 of our human peptide sequences could also be identified in the mouse database. If we compare it to a slightly larger, less stringent database that also contains predicted proteins, another 36 disappear. And if we use a custom database by using SNP polymorphism information in an NSG background, where we use this information to generate custom protein sequence databases, which is a very large database of over 350,000 sequences. An additional 42 of these peptides could now be mapped to the intermediate space of human-mouse conserved peptides. Okay, after all this, these bioinformatic analysis, we finally go towards selecting our peptides for the development of PRM assays. So here you can see it. So in these two quadrants that we are most interested, 493 glycopeptides found in serum and tumor, 1,900 that are found only in tumor. And the reason we were also interested in these, we call them kind of our tier 2 candidates, is all the problems that are associated with serum proteomics, which is huge dynamic range, poor sensitivity. So we reasoned maybe we should take peptides from here as well, maybe we have missed them in our discovery approach. Maybe we can nevertheless still quantify them if we use a more sensitive targeted proteomics approach. Okay, now mapping species, human unique peptides to these quadrants resulted in 257 human unique N-glycopeptides found in serum and tumor tissue, and 851 found only in the PDX tumors. We then required also a number of biophysical properties for these peptides that suggest one could build PRM assays. And these biophysical properties were as follows. So we wanted only fully tryptic peptides without any missed tryptic cleavage sites. We required a peptide to only have a single N-glycosylation site, and we required a peptide to be smaller than 25 amino acids, and that's for financial reasons. Synthesizing larger peptides as standards is extremely expensive. So that gave us 169 peptides mapping to 146 gene products for that set of peptides found in serum as well as tumor. And it gave us way more than 223. So using these biophysical criteria actually gives results in about 500 peptides. So here we have to be a little bit more reasonable, since it's quite expensive to synthesize stable isotope label standards, even if they're not ultra purified. So what we ask here, if multiple peptides mapped to the same gene product, we only selected the most intense peptide mapping to that gene product. That resulted in 223 peptides, then obviously mapping to 223 gene products. So it's one peptide per gene product. And nevertheless then covered every gene product in that list of 851 human glycopeptides. So in total, that's 392 peptides. We added 2 invertase peptides that we also synthesized as stable isotope labeled peptides. So this goes back to our internal quality control. So these were synthesized as bulk isotope labeled, stable isotope labeled unpurified peptides. So that's somewhat financially reasonable. One could do aqua peptides here, but would be extremely expensive. We then analyze these synthetic peptides by shotgun proteomics, and unfortunately some of our peptides contain methionine. One can't be completely selective if one follows N-glycopeptides, or peptides that come from post-translational modifications. So in total, 50 of our peptides contained an oxidized methionine, and 7 actually contained 2 methionines that were oxidized. Often this is likely substoichiometric. Nevertheless, we added these to our PRM targeted assays. So that then gave us a total of 451 assays that we needed to follow, light as well as heavy. So that makes up for 902. On top of that, we added 10 IRT peptides. So these are peptides that are used to measure the quality and reproducibility of your chromatography. So in total, 912 PRM assays that needed to be quantified and developed. Okay, that's how we designed the assays, or developed the assays, and I'm not going into absolute detail. So 912 events, we now developed them on a Q-Exactive HF. We selected a 90-minute gradient, so significantly shorter than what we had in our discovery approach, which was a 4-hour gradient. And then we had relatively short ion injection times, 80 milliseconds, and that's just one needs to optimize for speed here. So a lot of events that need to be quantified. Your cycle times would get out of control if one has longer ion injection times, which undoubtedly would make the assay more sensitive. We then used our stable isotope labeled synthetic peptides, and we developed assays using both first normal human serum as well as ascites samples from some high-grade serous ovarian cancer patients that we had available. So the reason for doing this, we didn't want to develop our assays in the valuable clinical samples, but we wanted to use our synthetic standards and samples that have the same, at least in theory, same kind of complexity as a background to develop our assays. And here's an overview of what we did. So here you can see on a chromatogram where on the x-axis is retention time, so it's 90-minute gradients plus 20-minute wash, so that's why it's 110. And on the y-axis you can see the number of concurrent PRM events that need to be quantified at that given time of chromatographic elution. So you can see sometimes we have to really follow a large number of peptides that co-elute at that time. If we use the synthetic peptides and we estimate what's actually the elution time, the average elution time of each peptide as it elutes from the column, this is probably not readable here, but it's a dendrogram where on the x-axis is peak start to end in seconds, and on the y-axis is number of peptides that have that elution profile. So on average, our peaks are about 45 seconds wide, from start to end, not half width. In the next chromatogram shown here in blue is estimated cycle times that this would result in. So since we're targeting a lot of peptides, sometimes cycle times here, even with the short ion fill times, can actually get quite long. Nevertheless, you know, with a peak that's about 45 seconds wide, that still gives enough data points to properly plot a curve for quantification. Here now you can see we also optimized normalized collision energies for each of these 451 peptides, and we did this by shotgun proteomics. So on the top of this slide, where you can see retention time versus intensity, every dot is a peptide that elutes. And then we didn't optimize collision energy by infusion, but by a shotgun proteomics experiment. So here these red dots, for example, would be the peptide that's indicated on top of that slide. So and we did about 6 or 8, probably 8 different conditions of different normalized collision energies shown in this graph on the right-hand side below on the bottom. So first one can see in red these peptides elute in a tight chromatographic window, and then choosing different collision energies in a stepped manner, we were able to select a collision energy that gave the most intense fragmentation, suggesting getting the most sensitive assay. And then here on that heat map on the bottom of the slide, we performed now scheduled PRM assays for all 451 heavy peptides that we spiked in approximately different concentrations from 25 to 150 femtomole on column. And one can see that this generates a linear response curve for each of these peptides, suggesting we can actually develop a PRM assay for every single one of these 451 targets. That, of course, as a word of caution, doesn't mean you can actually quantify them later in your clinical samples, but at least suggests we can develop the assay. And here now, you can see we now applied these 451 assays to a first set of longitudinally collected high-grade serous ovarian cancer serum samples. So we had serum samples from 10 patients and 5 longitudinal time points. At diagnosis, following surgery, at the end of chemotherapy, in remission, and in cancer recurrence. These patients all had clinical CA-125 measurements, and one can see this ELISA measurement in that graph on the right-hand side. We then performed our scheduled PRM assays using 1.5 microgram of serum glycopeptides. We spiked approximately 25 femtomole of heavy peptide, spiked the RT peptides, spiked 2 picomole of full-length invertase, and ran these in PRM duplicates. So while it's only 10 samples, that's still, if you add it up, that's 100 runs that had to be done. So significant mass spec time. Okay, and here's some quality control metrics. So here again, on the left-hand side, you can see the ELISA data for CA-125 in these 10 patients. On the right-hand side, you can see these are 100 runs. Each dot is a run. So on the x-axis, you can see the individual samples. On the y-axis, you can see the 10 IRT peptides, and then the 2 invertase peptides indicated by the arrows that we also had as synthesized as synthetic standards. You can see it's pretty reproducible chromatography over 100 runs. At the very end here, you can see we had a LC failure and a column failure. So that's where these kind of slight bumps actually happen, and one has to kind of reschedule these assays then. And these two samples indicated in blue, they did not behave well there. We had spray issues. They had to be removed. So it's about a 2% failure rate over 100 runs, which I think is reasonable. And then we quantified our 451 targets over these 100 samples. So first we averaged the technical replicates, and then we mined our data, making use of slope profiles based on ELISA for CA125. And one can see this here as a cartoon on the right-hand side. Each of these colorful dots on the bottom is a different time point for these longitudinal serum samples. So green is at diagnosis, purple is following surgery, blue at the end of chemotherapy, orange in remission, and red in recurrence. And if one looks at CA125 ELISA data, what's very obvious is that CA125 is highest at diagnosis, lowest at the end of chemotherapy. CA125 then actually stays kind of flat. And this is probably where the relatively poor sensitivity of it comes into play. So we asked the question, do we have any peptides that rise already in this stage? So they rise from complete chemo complete during remission and then keep further rising in biochemical remission. So for the first 3 time points we had, so basically highest at diagnosis, lowest at the end of chemotherapy, we had 148 candidates that had this decreasing kind of slope profile. We then had an additional 27 candidates, new candidates, that are rising at these time points. And then we added also invertase, and we also added the 3 positive controls that I indicated before, C125, mesothelin, and folate receptor. They actually were not rising, and I'll show you this on the next slides. Nevertheless, we kept them as positive controls. And here you can actually just see the raw data for these 32 candidates that I indicated. So this is plotted as a ratio of the intensity at diagnosis. So you can see highest at diagnosis, then goes down to chemo complete, and then rises again. And each individual peptide has a different color here. Okay, now look at some of the raw data. So first, let's look at CA125, which is the currently FDA-approved marker. So that's just a screenshot from Skyline. On the bottom, you can see the isotope, stable isotope labeled standard. On the top, you can see the endogenous, so the light channel that we quantified. And then this is indicated also in these bar charts on the bottom. So what you can see, the heavy spike in is relatively constant. The light is detectable at diagnosis, goes down after surgery, goes further down, but then we can't actually pick it up anymore. So that suggests, a little bit disappointing, but that suggests that CA125 PRM in its current condition is not as sensitive as an ELISA that has been optimized for 32 years. Maybe if one thinks about it, it's not completely surprising. Nevertheless, there's 27 candidates, as I indicated, that have these rising slope profiles that we were interested in. So that's shown here for this candidate X. I'm not showing you what it is on the bottom. So again, in the peak integrals, on the bottom it's the heavy peptide, on top it's the light peptide, and the bar charts actually show you again, on the bottom would be your stable isotope labeled peptide, on top would be an endogenous candidate. And again, you can see highest at diagnosis, goes down after surgery, goes further down at the end of chemotherapy, but then comes up rising rapidly again. Suggesting after validation, this could be a marker that's more sensitive than the currently used CA125. Okay, so as I said, now we narrowed this down to a much smaller number of candidates, and then we went into a second cohort of longitudinal samples. So now we have fewer targets. We can spend a lot more time of optimizing and tweaking parameters, which is not really feasible if you have several hundred. 12 additional samples, longitudinal samples, again 5 time points, again they have clinical CA125 measurement by ELISA, and again we run them in PRM duplicates. Okay, so the parameters that we then decided on optimizing. So now it gets pricey. So now we got highly purified and accurately quantified aqua peptide synthesized, light and heavy actually, for each of these 32 candidates. We optimized LC gradients, potentially making it also shorter, so we would have more throughput in the future. Of course, we rescheduled our PRM windows. We optimized ion injection times. I won't go into that, but we have longer ion injection times, which will then also increase sensitivity of these assays, as we could show. And then we generated response curves to show linearity and calculate level of quantification and level of identification for each of these peptides. So here you can see how we optimized the LC gradients. So first we ask what would be a good length or time for our LC gradients. Obviously, we would like to have them as short as possible. This gives us the best kind of throughput. So what we decided on is comparing a 30, 60, and a 90-minute chromatographic gradient that always contained 20-minute wash at the end. And you can see on top 30, in the middle 60, on the bottom 90. And then again on the right-hand side, one can see dendrograms of peak width start to end. So this is kind of following theory, right? If you have a shorter gradient, your peak width is more narrow. The peak is nevertheless more intense, suggesting you could have the most sensitive assay, but you don't have enough time to collect that. You have less time collecting data points over the elution of this peptide. So here we took a compromise and took the 60-minute gradient. Then we optimized gradient composition. So this is 0 to 20% B, 0 to 30% B, 0 to 35% B, and 0 to 40% B. And again, one can see how these 32 stable isotope labeled peptides elute over these chromatographic conditions. On the right-hand side, one again can see kind of the start to end peak width in seconds for these peptides. And on the next slide, one can see this plotted slightly different. We're on the x-axis, retention time of a peptide in a given gradient, and these gradients are indicated in different colors, so they're just different compositions of B, and they're compared to the 0 to 40% acetonitrile gradient. So what one would like to see in a plot like this is that these peptides form a nice linear diagonal. So you can see all of them do it, and you would like to make use of the entire gradient. So the 20 and 30% gradient, so the yellow and orange, they're not good. You can see peptides have not completely eluted over the analytical gradient, but they all come out at the same time in the wash steps. So based on these data, we chose the 0 to 35% gradient in a 60-minute elution window plus the 20-minute wash also. That gave us the nicest linear separation of these peptides, reduced chromatographic length already by 50% compared to our first stage, and also had peaks that weren't too narrow, so we still have enough time to collect enough data points over the elution of these peaks. And here you can actually now, again, kind of on the y-axis, in the x-axis, elution time, and on the y-axis, how many concurrent PRM events we're following at that given time. So if you compare this back to our initial stage where we had 450 and sometimes had to follow 60 targets at a given time, this is now dramatically less. So a max of 5 peptides at a given time had to be followed. So that gives us much more time, actually, we can sit on these peptides. Here we then used these synthetic peptides to generate response curves for every single one. The gray line is the calculated LOQ, and here I'll just zoom into one. So this keeps going much further. Of course, I only show you from 10 femtomole on column down to 0. Can see nice linearity, and based on this we can calculate the lowest level of quantification is 0.26 femtomole per 2 micrograms of serum on column. So it's a relatively sensitive assay. And here you can see again a couple of skyline screenshots for the raw data. So in this first slide, I would just like to show you actually invertase data. So that's kind of our quality control. Again, on the top is the is the light. So this is an invertase peptide that comes from the full-length invertase that we spiked in at the very beginning. And on the bottom, you can see the stable isotope labeled synthesized aqua peptide for this. The bar charts on the bottom show you the intensity for light and heavy, so it's relatively reproducible. And that scatter plot on the right-hand side shows you the variance. So there's about a 20% CV on these peptides. So that's reasonable considering that's a relatively lengthy isolation protocol, enrichment protocol for these N-glycopeptides. And then here again, let's some of the candidates that I showed you in the first round, in the first serum cohort before. Again, on top light, on the bottom heavy, and here the box plots, they actually ratio. So again, you can see this particular candidate 1, has the highest abundance at diagnosis, goes down after surgery, goes down after chemotherapy, and rises up again in remission and recurrence. And again, there are peptides, candidate 2, candidate 3, that follow that same trend that we now could already validate with a more sensitive assay in a new cohort of samples. And we're just in the process of finishing the analysis of all these 12 samples with all the 32 candidates. So in summary, so we used primary patient-derived xenograft models and a chemical proteomics approach to specifically enrich for N-glycopeptides. These are peptides of high relevance since most current FDA-approved biomarkers are N-glycoproteins. These PDX models enable us to assign species to peptides that we identify, and hence unambiguously identify peptides or proteins that are made by the tumor, released from the tumor, and detected over serum background proteins using current shotgun proteomics approaches. We then synthesize a large number of these peptides as stable isotope labeled standards and systematically build a large number of PRM assays that we initially apply in something that we call a PRM screen to cohort 1 of longitudinally collected patient serum samples. We then mine these data based on slope profiles, narrow it down to a much smaller number of 32 peptides. We synthesize these as aqua peptides and apply to a second cohort of samples, and we're just in the process of completing these analysis. So here it's just an outline. So all this work actually was done by an excellent grad student in my lab, Ankit Sinha, in a one-man show, so to say. He had some help from two bioinformaticians in my lab, Vladimir and Alex, and we had some great collaborators that either developed these xenograph models, Ben Neal and Laurie Ailes' lab, or that provided us with the clinical serum samples, Marcus Bernardini and Blaise Clark. And this project was funded by CRHR in Canada. And thank you for listening, and I'm happy to take questions from the audience.
Thank you, Dr. Kisslinger, for walking us through your important research on identifying peptide biomarkers indicative of ovarian cancer recurrence. For our attendees, this is your last chance to submit a question. If you'd like to submit a question, simply type it in the box to the left and hit submit. A reminder that today's webinar will be available on demand in 72 hours should you want to listen again or share it with a colleague. Thank you all for attending today. Please bear with us as we compile your questions for the Q&A. We'll be back with you shortly. Okay, we've had a few questions come in over this presentation. Thomas, again, thank you for an interesting presentation this morning. And let's get to the questions. So the first question, Thomas, is what is the engrafting take rate for the IP PDX models compared to the mammary fat pad PDX models?
So as I mentioned in the presentation, so these PDX models were originally developed by Ben Neal and Laurie Ailes' lab, as published in this PNAS paper in 2011. And here, viable cells were engrafted into the mammary fat pad of NSG mice, and the take rate was very high. It was about 95, almost 100%, 95%. So when we then reestablished these PDX models by taking viably frozen single-cell suspensions from surgically resected high-grade serous ovarian cancer patients and IP engrafted them into the same strain of immunocompromised mice. Take rate is much slower. Is much lower. It's about 50%. And also the growth kinetics of these tumors is significantly slower. So it takes about half a year or so for these animals to develop these kind of advanced stage high-grade serous ovarian cancer type phenotypes.
Okay. The next question that we received is, were the PDX models characterized by genomics? And were they from specific molecular subtypes of high-grade serous ovarian cancer?
So as I mentioned at the very beginning of this presentation, epithelial ovarian cancers present in 5 different histological subtypes, and high-grade serous ovarian cancers are the most common subtype among ovarian cancers. And sequencing studies, and now even proteomic studies, so sequencing studies from the TCGA or proteomic studies from CPTAC have shown that these high-grade serous ovarian cancers exist in several different molecular subtypes. So when this original paper, again by the Neal group, was published, they certainly did lots of genomics, both on the primary as well as on the PDX tumors. So that data is available, and they could also show that based on copy number or based on exome-seq, I think, as well as RNA-seq, these PDX models nicely recapitulated these molecular subtypes of the primary tumors. So we, in our cases, since the numbers were relatively low, did not redo the genomics, and we also did not select specifically for molecular subtypes of these primary tumors prior to engraftment. The numbers just weren't there, so this didn't make any huge sense at this stage of the project.
Okay, very good. Just a reminder to our attendees that if you would like to ask Thomas a question, simply type your question into the Q&A box on your screen and hit enter. Okay, so the next question we have is, where were the isotope label standard peptides purchased from? And for the PRM-MS screen, were they accurately quantified?
Okay, so we actually, we had 2 stages of PRM development. So in the stage 1, where we had these 400 or so, 451 candidates, obviously we could not afford to buy accurately quantified aqua peptides. So we bought stable isotope labeled bulk synthesized peptides that were not accurately quantified. So that, of course, doesn't then allow you to do this quasi-absolute quantification. You can really only do a relative quantification, and these peptides were purchased from JPT. And then as we narrowed down our number of candidates, then we actually purchased, highly purified, accurately quantified, in solution, stable isotope labeled peptides from Thermo Scientific, so the aqua peptides.
Okay, very good. And are all candidates detected in every longitudinally collected human serum samples?
So that's a great question. So of course there's variability. So these views of raw data that I showed you, not every peptide in every patient will show you these type of slopes or response curves. So the answer is no. So they were not detected in every single patient that we analyzed. And, that might be expected. I mean, CA-125 is also not detected in every single high-grade serous ovarian cancer patient. So, and that's where likely signatures will come into play. So our goal now is, once we finished this final set of 12 patients, can we come up with small peptide signatures among these 27 new candidates that would capture the entire cohort of patients? And then move from there into the next stages of this project.
Okay, thank you for that. We have a question that's come in. I'm not sure if it's something you want to take a swing at, but it's more in the genomics realm. But what do you think about the TERT gene as a marker for early ovarian cancer diagnostics in human blood samples? FISH assay or NGS? I mean, I know you're doing proteomics work, but...
Okay. So, I mean, interesting, I've not heard of this genomic marker. So, as you can imagine from my presentation, we're a proteomics lab and not a genomics lab. So, I cannot fully answer if this marker would be good for early detections. I mean, early detection in high-grade serous ovarian cancer patients is a tricky business, right? So, I mean, as you saw at the very beginning of my presentation, these tumors likely originate in the surface epithelium of the fallopian tube, and then very early transformed cells or clusters of cells disseminate into the abdominal cavity. So at that stage, to a certain degree, one can almost call it a metastasis. So one would almost need to detect it at that very early stage. So I'm not sure if a blood test with any type of molecule, DNA, RNA, or protein, will be sensitive enough. So I've seen projects that look at vaginal flushings and looked at ctDNA or circulating tumor cells. That might be something that could be done. So how this would be implemented into the TERT gene, I do not know the answer to that.
Okay, very good. We have one last question in the queue for you. It's pretty straightforward. And so based on where you've come with your most recent work, what would be your next steps for this study?
So, yeah, also a very good question. So obviously, I mean, it took several years to get to this stage, right? So it was kind of a real tour de force, which, you know, made use of nice model system, PDX model system, and some bioinformatics tricks to identify these human-secreted peptides that could potentially use as tumor volume or biomarkers of early detection of recurrence. I mean, but what and of course there were lots of technical considerations to this paper. How do you build PRM assays of sufficient quality for such a large number of candidates, and how you narrow them down so you can actually further optimize these assays. But what you could, of course, see, I mean, the number of patient samples were still relatively low. So 10 in this first stage longitudinally, so that makes up 50 serum samples, nevertheless, and then 12 in the second stage, so that makes up 60 serum samples. So it's not that it's a totally tiny number of individual samples, but it's a relatively small number of individual patients. So what we are interested in, of course, now is narrowing it down to these small signatures. Maybe we can even we can narrow it down even further from these 27 candidates, maybe to a small peptide signature of 5 to 8 peptides, which we could then further optimize and hopefully reduce chromatography gradients and get more sample throughput. And then, of course, we would need to apply this to a much larger cohort of longitudinally collected serum samples from these ovarian cancer patients to see if this holds up. So that really would be the immediate next steps, narrow down to a signature, further optimize the assay conditions for these signatures, and apply to a larger cohort of patients, probably in the hundreds of patients, to be really properly powered.
Mm-hmm. Excellent. Well, as I see there are no other questions in the queue, I think it's time to wrap up today's webinar. Thank you very much, Thomas, for your time in the presentation today on your latest research using mass spec to identify peptides for the detection of ovarian cancer recurrence. I would also like to thank all of our attendees for joining us today. And as a final reminder, you can access a recording and a replay of this webinar 72 hours from our closing time. Thank you, everybody, for your participation today, and have a nice day.
Thank you from my side as well.